Hi, good morning. My name is Gabriela Sanchez. Today is Saturday, November 14th, 2020. Today I'm going to be doing the video for microscopy for microbiology, and I'm going to be going over the different parts of the microscope. I am currently using the HOL V-Scope 1500. And so this only has one eyepiece. So I'm going to go through the whole process, or all of the parts. This here would be the ocular lens or eyepiece, and this pretty much allows you to view the slide. So this is where you put your eye. The arm is this part over here. This pretty much supports the lens of the microscope, and it connects it to the base of the microscope. The turret is this part here, and that could be switched around. It really lets you choose the proper magnification power that you need depending on which slide that you are seeing. The objective lens, which is, so the turret is this part, the thing that kind of swivels back and forth. The objective lens are these three. Those pretty much focuses on the specimen. So that's what these are. The condenser lens aligns the focus lamp onto the specimen. The stage would be this part here, and the clips are here. The diaphragm sits right below here, and it alters the projected, the amount of the light projection up to the condensed lens, which is up here. The illuminator is down here. This is the light source. It switches on and off. And the coarse focus knob, as well as the fine focus knob, are here. So this is the coarse focus knob. This kind of brings the object into focus by greater quantities. So, I mean, it makes a more drastic change. The fine focus knob would be this one. Those are like for real fine adjustments that you want to see when you are ready to look at your slide. The transverse stage control knob, are these two. So the transverse and the longitudinal stage control knob, they are used to either move the slide back and forth or from side to side, and it moves this, not the stage, but it kind of moves the slide back and forth or up and down. The base is this bottom part, and it's just to kind of support the microscope. I think I went over all of the parts of the microscope. I have already viewed all of the slides that were provided to us in our lab kit. So I have the yeast homel, the bacteria spirillum, bacteriococcus form, the letter E, focus, bacteria's bacillus form, penicillin with conidia, and amoeba with whole mount. So all of those I've already looked through the microscope. I've also already created and viewed my wet mounts of the cheek as far as the dental plaque, and I saw those under the microscope as well. The way that I did my slides was that I grabbed a sterile like Q-tip thing, and I vigorously swabbed the inside of the cheek for the wet mount of the cheek, and then vigorously swabbed the dental area right by the gum line vigorously to obtain samples from those. I placed it on the slide, and I've already discarded them, so I apologize. I applied the cells on the slide in a circular motion. I added 1 drop of distilled water and covered at an angle with the cover glass. So I did it at an angle so that I was able to get a flat surface, and if there was any excess water, I just damped it with the paper towels. I looked at those under the microscope, and I was able to see the different cells of the cheek and the dental plaque. I will make a follow-up video on what I saw on the next video. So that concludes my video for the first part. Thanks.